Dear students, myself Dr. N. Ramakrishna, Assistant Professor in Botany, and my colleague Dr. Y. Kumar is also Assistant Professor in Botany. So, today we are going to discuss, we are going to deliver in entire the algae life histories, some of the members of algae life histories. So, that is Oedogonium, and the second one is Sargassum, and third one that is in Polysiphonia. Sir, what is the meaning of in algae? How many classes in algae? What is the life histories of in algae, sir? Please.
Algae definition, they are the thallophytic autotrophic, simple plant, that is a simple definition. Thallophytic means there is no differentiation in root system and shoot and leaf.
Okay, sir.
So, that is thallophytic, and they can prepare their own food material. So, this is about algae, and in algae there are 11 classes are there, and if you take some of the classes, important one, chlorophyceae members, phaeophyceae and rhodophyceae, that we are going to discuss. From chlorophyceae member Oedogonium, and phaeophyceae member Sargassum, from rhodophyceae Polysiphonia.
Sir, how can we recognize in chlorophytic member, sir?
Chlorophytic member recognized based on the chlorophyll pigment,
Okay.
chlorophyll A and B, that is chlorophyceae members.
Okay.
Chlorophyll A and C and some of the xanthophyll pigment, that is phaeophyceae,
Okay.
brown in color.
So, now we are going to discuss in chlorophytic member in Oedogonium.
Yes, chlorophytic member Oedogonium, and phaeophytic member Sargassum,
Okay.
and rhodophyceae member Polysiphonia.
Polysiphonia. Okay, sir. Sir, how can we recognize in Oedogonium? What is the cell structures, and what is the sexual reproduction system? What is the life history, sir, please?
Okay. Actually, we can recognize based on the thallus structure. If we take thallus, Oedogonium thallus, the Oedogonium thallus, unbranched filamentous thallus, the cells arranged one above another. The cells are, if you take each cell cylindrical elongated structure. So, based on this we can identify it, and apart from that there are so many unbranched filamentous there, but especially in Oedogonium there are polarity. The thallus shows the polarity. Polarity means we can recognize here the basal cell hold fast and apical cell. That is the one of the characteristic features. And another one is capsule present in the thallus.
Okay.
So, this is the recognition
Sir,
character.
what is the differentiation between hold fast and vegetative cells, sir? How can we recognize in hold fast and vegetative structure, sir?
Hold fast only, that is the function is fixation.
Okay.
And the vegetative cell, this is elongated cell and cylindrical structure, and it perform the photosynthesis, but not perform, the hold fast not perform the photosynthesis because of very less chlorophylls present inside the hold fast.
Okay, sir.
That is the different.
Yes. So, what is cell structure of Oedogonium?
If you take thallus, not only thallus, any organism, it is made up of cell. So, like that the thallus also made up of cell, that is the structural unit. Cell is the structural unit. If you take cell in generally, the cell components are cell wall and protoplasm there. In protoplasm, cytoplasm, number of cell organelles are there. So, like that here also same, the cell composing of cell wall, and inside the cell the cytoplasm with nucleus. We can find out the clear nucleus, and also all cell organelle, all eukaryotic cell organelle, and especially chloroplast present. And if you take the cell wall, in cell wall it is made up of clearly 3 layers: outer chitin, middle pectin, and inner cellulose. And the important cell organelle is chloroplast. In Oedogonium, chloroplast is reticulate chloroplast. That is also one of the characteristic feature of the Oedogonium. And this reticulate chloroplast with number of pyrineads, we can observe in diagram. These are the pyrineads, and this is reticulate type of net-like chloroplast, and this is the nucleus. And inside the chloroplast, pigments are there, chlorophyll A and B, and xanthophyll and keratins and carotenoids, they are all present inside the chloroplast.
Sir, what is the main functions of in pyrineads, sir?
Pyrineads, actually the pyrineads have the one of the important enzyme, that is the Rubisco enzyme. Okay. The function of Rubisco enzyme, they promote the photosynthesis, and due to the photosynthesis food material prepared. This food material stored inside the pyrineads.
Okay. It is only food material prepares the pyrineads.
This food material may be stored inside or outlayered.
Okay, okay, sir.
The mostly the food material in starch, outlayer of the pyrineads.
Pyrineads, yes.
Reproduction.
So, what type of reproduction system in Oedogonium?
The Oedogonium, there are 3 types of reproduction: vegetative reproduction, asexual reproduction, and sexual reproduction. If we take vegetative reproduction takes place by fragmentation method. That means in name itself given vegetative, if any vegetative part separate from the filamentous thallus, they can grow as a new filament. So that is the fragmentation process. And another important one is in Oedogonium, asexual reproduction. If we take asexual reproduction, this asexual reproduction takes place by formation of special type of juice spores. These special type of juice spores called as Stephanocanthe juice spore. Let's see how the juice spores formed inside the Oedogonium. The first of all, this is the Oedogonium thallus. The Oedogonium thallus made up of number of vegetative cells. This vegetative cell, vegetative cell, vegetative cell, and these cells already we discussed in cell structure. It is covered by cell wall layer. Except the cell wall, rest of the portion called as protoplast. Before formation, the spores, what happened here? This protoplast, the cell protoplast, separated from the cell wall and round up and metamorphosed as a spore. And this is actually this vegetative cell. The first this vegetative cell enlarged in size. Now it is called as a sporangium. After sporangium formation, the protoplast separated from the cell wall and round up like this. We can see in C diagram here, round ups. And before formation spore, what happened? The hyaline colorless structure formed at the apical region here, and this hyaline structure surrounded by the number of flagella. This number of flagella at the one region. So this special type of spore called as Stephanocanthe. This is also one of the characteristic feature of Oedogonium. After formation spore, this Stephanocanthe spore inside the sporangium, the sporangium wall rupture. As a result, this spore released out and takes sufficient place and falls on sufficient place at the region of hyaline side, colorless hyaline side. Then immediately the flagella shed off. We can see in C diagram flagella present, but here in D diagram flagella shed off. Later from the hyaline side, the whole flask developed and rest of the cell developed as a vegetative cell. Like that the formed. This is about asexual reproduction.
Sir, this asexual reproduction system, it is an unfavorable condition or favorable condition for
yes,
formation?
Good question. Actually, the spore formation mainly the favorable condition.
Okay.
And unfavorable condition also spores form.
Yes. Sir, this spores comes to when it comes to outside the sporangium, automatically it is formation in the new thallus.
Yes.
Yes. Okay. Okay. Sir.
Formation inside the sporangium.
Okay.
But after form the spore, they are released out.
Okay.
After release, take sufficient place. They can germinate as a new thallus.
New thallus.
Right.
Okay. But unfavorable condition it is. Unfavorable condition.
Spores favorable condition.
Okay.
But in an unfavorable also takes place, but the formation of actinids.
Okay, sir.
The actinids means the hard cell wall layer.
Yes, yes.
That is actinids, unfavorable actinids. The favorable spores.
Yes. So this is total asexual reproduction system.
Yes, this is asexual reproduction.
Yeah. Sir, what is sexual reproduction system in Oedogonium, sir?
Sexual reproduction takes place by formation of gametes, male gamete and female gamete. Let's see the sexual reproduction process. So definition clear, the fusion of male and female gamete, that is called sexual reproduction. And commonly the male gamete developed inside the male gametangium and female gamete developed inside the female gametangium. Here the male gametangium are called as antheridium, female gametangium called as Oedogonium. Right. So like a formation spore, the gamete formation also same. The same if you see in this slide, the first vegetative cell before formation gamete, first vegetative cell, this vegetative cell metamorphosed as a antheridial initial when it is enlarged in size. After that antheridial initial developed as a antheridium. Inside antheridium male gametes are formed. And the likewise same here the same vegetative cell, Oedogonial initial, Oedogonium and female gamete. This is formation. If you see the diagrammatically, so this is the thallus structure, Oedogonium thallus. The first single cell, the single cell divided and form number of cell. These are called as antheridial initial, and later it is developed as a antheridium one by one. This is antheridium. So after formation like this antheridium, inside the antheridium what happened? One mitotic division takes place. Due to the mitotic division, two anthrozoids are formed. two anthrozoids are formed. These are the anthrozoids from one antheridium. So these anthrozoids, another name male gametes, and after formation these anthrozoids or male gametes released out and swim in water and reach to the oogonium, where fertilized. As result, formation oospore, which is called as zygote.
Sir, here oogonium, it is a female reproductive organ.
Yes.
Okay, that one is antheridia goes to where the female reproductive organs.
Yes.
Yes, sir.
Antheridium male,
Yes.
oogonium female.
Female.
In antheridium male gametes develop.
Okay.
In oogonium female gamete develop. This type of sexual reproduction advanced oogamous type.
Okay.
Oogamous type means actually if we take the another type, here the male and female released out. Male gamete and female gamete both are released out and fused. But in oogamous type only male gamete released out, but not female gamete. The female gamete developed inside oogonium and situated inside the oogonium,
Okay.
but not released out. Now the already released male gamete fused with oogonium egg cell.
The female gamete. Okay,
Right.
sir. Okay.
As a result, formation of oospore are called as zygote, which is diploid state.
The Oedogonium, it is two type of species is there. Sir.
Right.
One is the Macrondus, second one is the Anandrus. So what is the differentiate between Macrondus and Anandrus? How can we recognize it in Macrondus and Anandrus, sir?
Now we discussed Macrondus type. In Macrondus type, what happened? The male gametes directly formed.
Okay.
But in Anandrus, this male gamete indirectly formed on specialized branch. The specialized branch called as dwarf branch. Let's see non-Anandrus species also. So far we completed about Macrondus species. If you see the non-Anandrus species, first, actually this male gamete, what happened in Macrondus? In Macrondus, this male gamete directly formed from the antheridium. But before that, in non-Anandrus species, what happened? The first vegetative cell, this vegetative cell developed as a androsporangium initial. Instead of antheridial initial, androsporangium initial. This androsporangium initial developed as a androsporangium.
Okay.
Later which produces androspores. After formation androspores, these androspores reach to the oogonium wall. Only oogonium wall or subaltary cell.
Okay.
Where it is divided and form the specialized branch on the wall. And this specialized branch called as dwarf branch. And the apical cell of the dwarf branch developed as a antheridium. And inside antheridium male gametes are formed. If you see the diagrammatically, this is common vegetative cell divided first of all this is androsporangium initial. Later it is developed into androsporangium like this, one androsporangium, these are androsporangium. From the androsporangium, these androspores are released out. Now this androspore reach to the oogonium wall, or some species they can fuse with subaltary cell also. But after attached to the oogonium wall, immediately what happens? The spores are shed off. Androspore flagella shed off, and immediately division takes place. As a number of cells are formed. The apical most cell, this is actually this total called a dwarf branch. The apical most cell, it is act as a antheridial initial. If we take here this antheridial initial, antheridium. Inside antheridium what happen? Inside antheridium mitoid division takes place like a Macrondus. Due to the mitosis division, two anthrozoids or male gametes are formed. So these male gametes or anthrozoids released out and now fuse with oogonium egg cell. As a result, formation of oospore or zygote.
Yes.
This is about non-Anandrus species.
Okay, means here we are looking two type of the species. One is
Yes.
Macrondus and Anandrus.
Yes.
Yes, sir. Sir, what is the life histories of Oedogonium?
If we take life history, first let's see the Macrondus life history.
Okay.
If we take Macrondus life history, so this is the Oedogonium thallus. One side asexual reproduction and sexual reproduction. I am taking only two sexual reproduction without vegetative. So here the first one of the vegetative cell developed as a zoosporangium initial. Inside zoosporangium initial protoplast round up and form as a stiphenocontate zoospore, and this stiphenocontate zoospore germinate as a again Oedogonium. There is no fusion of gamete. The spore directly gives rise to Oedogonium thallus, that is asexual. And if you take the sexual reproduction, here oogonium and this is antheridium. From antheridium male gamete released out. Already inside the oogonium egg developed, but the egg not released out is situated inside the oogonium. Now they are already released male gamete swim in water and fuse with egg cell. As a result, formation of diploid zygote or oospore. After formation diploid zygote, now it is undergoes to meiotic division. As a result, formation of spores, juice spores, again juice spores form. Now the juice spore takes sufficient place and again germinated as a Oedogonium thallus. Now this is life cycle. We start the cycle with thallus and again end with thallus forms.
Okay, this is the life cycle of
macrandus. If you take the non-androus cycle, the same thallus structure already asexual reproduction completed, same asexual reproduction in macrandus and non-androus, but sexual reproduction different. If you take sexual reproduction, first what happened? Here the androspore formed, androspore formation. After androspore, these androspores attached to the oogonium wall. After attachment to the oogonium wall, they are developed as a dwarf branch. On the dwarf branch, again male gametes are formed. Now this released male gamete fused with egg cell of the oogonium.
Okay, so that is the life history of macrandus and non-androus species. So this is of in chlorophytic member.
Yes.
Sir, next now you see one more most important member is that is a phaeophytic member, sir. How can we recognize the phaeophytic member? What is the importance of phaeophyty? So what is the member of in phaeophytic?
In phaeophyty, there are ectocarpus, number of species are there, but according to our syllabus, sargassum is important.
Okay.
So phaeophytes recognition actually based on the chlorophyll and some of the pigments, they are the brown in color because of chlorophyll C and pheoxanthin, mainly pheoxanthin. That's why it is in brown in color. And if you take the sargassum, another name is gulf weed. They are mostly found in oceans, in littoral, sublittoral region, tropical temperate zones. If you take the thallus, this thallus is macroscopic structure. If already Oedogony completed, the Oedogony not a macroscopic, Oedogony is a microscopic, but we can directly recognize sargassum based on without direct eye. We don't use any microscope.
It is seeing to in our eye.
Yes, directly.
It is a just plant-like structure is there.
Yes, it is like look like a mini plant.
Plant-like structure.
And also the plant-like parts also we can observe.
Okay, the Oedogonium only we are looking under microscope.
Yes.
It is not in our eyes.
Right.
Okay, that is a variation.
Right. The plant-like structure means we can hold fast part like a root-like, and stem-like. This is stem, main branches, and also leaf-like appendages also we can observe.
Okay.
That's why it is look like a mini angiospermic plant. Of course, it is algae, but look like a plant.
Looking like a plant. Okay, sir.
And these lateral leaves called as cryptoblast. And apart from that, some of the receptacles. These are the fertile branches. These are called receptacle. On the receptacle, conceptacle present. Inside the conceptacle, sex organs are developed. So this is about the sargassum thallus structure.
Okay. So what is the reproduction system of sargassum? So here, what type of asexual, what type of sexual, what type of life histories in sargassum, sir?
Mainly, only one type of sexual reproduction, majority, that is the sexual reproduction.
Okay. Advanced type reproduction
right,
system.
same advanced type oogamous, like a Oedogonium.
Okay, sir.
Like a Oedogonium, same advanced type oogamous type. The same like Oedogonium, here also same male antheridium, female oogonium. But here the difference is, this reproductive organ in Oedogonium, the reproductive organs develop from the direct vegetative cell, but the specialized branch are there. This specialized branch called as conceptacle. Inside the conceptacle, this is conceptacle in internal structure. If you take the conceptacle in internal, the antheridia developed in a cavity. These are antheridia. Apart from the antheridia, the protective hairs also developed. These are called paraphyses. So you know, after formation antheridia, inside the antheridia, male gametes are formed. This male gamete released out and reached to the female reproductive organ. In female reproductive organ, egg is present. So this male gamete fuse with egg. This process called as fertilization.
Fertilizations. Okay. So after the fertilization, formation is zygote.
Right.
Yes.
And another important one is, it is a sargassum. Actually, if you take the Oedogonium haploid. Haploid means it is only one set of chromosomes. After formation zygote, diploid zygote, the diploid zygote undergoes to meiosis and again formation haploid spores. But in sargassum, after formation zygote, no meiotic division. Now this diploid zygote undergoes to mitoid division and developed into diploid sargassum plant. That is the difference.
That is a variation of.
Right.
Yes, sir. So, what is the life history of sargassum, sir?
If you take sargassum life history, first in phaeophytes, in sargassum also, vegetative cell, antheridium initial. Apart from the antheridial initial, paraphysis also develop inside the cavity.
Okay.
And antheridium.
So, what is the paraphysis, sir?
The paraphysis is only the protective layers.
Okay.
The protective
so
layers.
So, where the reproductive organ.
Right.
Yes, sir.
Right. So, in antheridium, male gametes are formed.
Okay.
So, after formation, this male gametes released out as the same way. Here also same vegetative cell, Oedogonium initial and Oedogonium developed inside the Oedogonium egg cell.
Yes, female
now,
gametes.
the male gamete fused with female gamete egg.
Okay.
As a result, formation.
When the male gamete comes to outside that area, it goes to where the female reproductive.
Female, as it is a oogamous method.
Okay.
Female gamete not released out.
Male gametes are released
Only male gamete released out.
Okay.
Female gamete situated inside the Oedogonium.
Okay.
After formation zygote, mitosis division and again developed into thallus.
New plant. So, this is of sargassum, sir.
Yes.
Yeah. So, after next one, that is of rhodophytic member, sir. So, how can we recognize the rhodophytic member? What is the structure of in rhodophytic? Similarly, what is the sexual and sexual life histories of in rhodophytic member, sir?
Okay. Rhodophytic members mainly they are called as red algae because it consisting phycoerythrin, phycocyanin. Due to the phycoerythrin, these are called red algae. And apart from that, chlorophyll A and chlorophyll D also present. And in this, according to syllabus, the most important one is Polysiphonia type study. If you take the Polysiphonia, the name given Polysiphonia, poly means number of. Siphonia means
the
tubes.
plant body, it is a total pipe-like structure,
Yes.
tube-like structures is there.
That is called siphonous.
Okay,
Tubes in botany
Yes.
language siphonous.
Siphonous, okay, sir.
So that is called as Polysiphonia.
So,
That's
means
why,
many tube-like structures is there.
yes, many tube-like structures formed.
Okay,
That's why name given as a Polysiphonia. And if you take life cycle, the clearly 3 stages are there. One is gametophytic stage, haploid. Second, carposporophyte stage, diploid. And again, tetrasporophyte stage, diploid. This life cycle called as didiplantic life cycle because diploid stages 2 times. One is carposporophyte, second tetrasporophyte. First, let us see the gametophytic stage. So, in gametophytic stage, already the Polysiphonia plant body, that is gametophytic.
Okay.
After gametophytic plant body developed, when it is reached maturity stage, and when it reach maturity stage, the reproductive organs are developed. The reproductive organs, the male one called spermatogea and female one called as carpogonium.
Okay.
Let us see the spermatogea development. So, this is the from the tube cell, one of the cell developed into trichoblast, that is the fertile branch. After formation trichoblast, from the branch, the spermatia like this. These are all spermatia. The spermatia are developed.
Spermatia means it is a male reproductive organ, sir?
Yes.
Yes.
Like a male gamete.
Male gametes.
Non-motile male gamete.
Okay. Okay.
So far we discussed in Sargassum, Oedogonium, they are the motile male gamete.
Motile male gametes.
But in Polysiphonia, non-motile.
Okay,
So this is called spermatium. So after formation spermatium, they are released out from the spermatogea. This is cells spermatogea. From the spermatogea, spermatia released out. After release this spermatogea, these are actually due to the lack of flagella, they can move with help of the water current to the female reproductive organ. Let us see the female reproductive organ here, carpogonia. So this elongated neck called as it has the one elongated neck and swollen venter. Inside the venter, here the egg cells are present. Here egg is present.
Okay.
So after release of the spermatia reach the this neck-like part called trichogynia. Reach the trichogyne and fuse with egg cell or female gamete. As a result, formation of zygote. So this is only gametophytic stage. After formation zygote, now from the zygote to developed as a carposporophyte stage. Let us see the carposporophyte stage.
Sporophyte. From here to we are studying in sporophytic stage.
Yes. Now gametophytic stage end.
Okay. So you see, before we are two completed, there is no sporophytic stage, sir?
Yes, sporophytic stage, that is zygote itself sporophyte.
Okay.
There is no clear sporophyte.
Sporophyte.
In
So from
Oedogonium
here to
and
here
also,
to we are studying to sporophytic stage. Yes,
so
sir.
that is the one of the advanced character.
Ah, yes.
If
So
you take
this
the plant body, gametophytic stage and sporophytic stage are
sporophytic
there.
stage, from here to we are studying in sporophytic stage. This is the
Right.
advanced stage of in
Advanced stage.
Okay,
And most of
rhodophytic
the
member.
primitive plants, the main plant body gametophytic stage.
Gametophytic stage.
If you take the algal member, all algal member, that is the gametophytic stage.
Okay, okay.
And when it is reached to bryophyta, apart from the gametophytic stage is there, that is dominant.
Okay.
On the gametophyte, sporophytic different stage develops.
So alternation of generation is there
Right,
in bryophyte.
alternation generation.
Generation developed.
So main gametophytic, on the gametophytic, sporophyte dependent.
Yes, okay.
But when reach the pteridophyta, the both are separated.
Both are separated.
That means sporophytic stage separate and gametophytic separate. And sporophyte is dominate.
Yes, but here this is advanced character
Yes.
in algae group.
Yes. Here gametophytic stage dominate.
Okay.
And later sporophytic stage also develop.
Starting stage sporophyte development
Yes.
stage. Yes, sir.
So anyway, after diploid zygote formation,
Okay.
this diploid zygote divided, and one after division, two nuclei formed. One of the nuclei entered into the axillary cells, where here gonimoblast filament formed.
Okay.
After entering into the axillary cell, where the gonimoblast filament formed, and in this gonimoblast filament, the apical most one called as carposporangium.
Yes.
Inside the carposporangium, carpospores are formed.
Okay.
That entire this structure called as, the part called as cystocarp.
Cystocarp.
This stage called as a carposporophyte stage, as
Yes.
it is diploid carposporophyte stage. So anyway, after formation carpospore, inside the carposporangium, these carpospores are diploid. Now after mature this carpospore, these carpospores released out, and now it is enter into the next generation, that is the third one, that is tetrasporophyte stage. The tetrasporophyte stage also diploid. Let us see tetrasporophyte stage. This is tetrasporophyte.
Okay.
So inside this thallus, the cell developed as tetrasporangium. You can observe in diagram. In tetrasporangium, tetraspores are formed.
Okay.
These tetraspores are developed by meiotic division. Due to the meiosis, haploid tetraspores are formed. So these haploid tetraspore further developed into gametophytic thallus. Polysiphonia, gametophytic thallus. Now the class start with gametophytic thallus, gametophytic stage. Later the second stage carposporophyte stage. After formation zygote, the zygote developed as a carposporophyte. In carposporophyte, the end carpospore released out, and these carpospores again developed as a tetrasporophyte. Now the tetrasporophyte developed as a again gametophytic thallus.
Okay. So four stages is there in this one.
Mainly three.
Three stages is there.
Gametophytic stage, carposporophyte stage,
Yes.
and tetrasporophyte stage.
Okay, okay.
Let's see the life cycle. Here first gametophytic, male gametophyte, it is dioecious.
Okay.
Male separated and female separated. The male produces spermatangia, female carpogonia. The spermatangia, spermacia develop, and carpogonia in swollen structure egg or female gamete. Due to the fertilization zygote, the zygote developed as a carposporophyte. In carposporophyte, carposporangia produces carpospore which are diploid.
Okay.
This diploid developed into tetrasporophyte. In tetrasporophyte, tetrasporangium developed. In tetrasporangium, due to the meiosis division, haploid tetraspores formed. This tetraspore again developed as a male and female gamete.
gametophyte.
So this is Polysiphonia.
Life history. So
Life
here the most important carposporophyte and carposporangia and carpospores.
Yes.
Yes. So okay. What is the importance of in this rhodophytic member?
Rhodophytic member from the cell wall, carraginin, agar-agar, they are extracted from this rhodophytic members.
Sir, what is the differentiated between Oedogonium Sargassum and Polysiphonia?
In terms of thallus, Oedogonium unbranched filamentous, Sargassum, it look like a miniature plant. Polysiphonia, it is made up of number of tubes. That is in thallus wise. And if it is reproduction, in Oedogonium, the specialized juice pores are formed. They are called as a stiphenocontate juice pore. This type of stiphenocontate juice pore is only found in Oedogonium, but not in Sargassum and not in Polysiphonia.
Okay.
And if you take the sexual reproduction, in Oedogonium, there are two different type of species. Macarondas and Anandrous.
Okay.
And formation Anthridium, Oedogonium, in any species, whether it is Oedogonium, Sargassum and Polysiphonia, any one, the male reproductive organ Anthridium,
Okay.
and female one is Oedogonium or female gametangium.
Yes, yes.
But here the formation different. If you take the Oedogonium, vegetative cell itself developed as a reproductive organ,
Okay, okay.
like a Anthridium and
Oedogonium.
And if you take the Sargassum, in Sargassum there are specialized branch. There's a conceptual specialized branch there.
Special structures is
And
there.
in specialized branch, the sexual organs deposited inside the concepticle.
Okay, okay.
And here also same Anthridium, Oedogonium.
Oedogonium.
But
So
that
similarly,
was somewhat complex.
but rhodophytic also some
Paraphysis
complex
form.
structures is
And-
there rhodophytic also.
And if you take the rhodophysis member, Polysiphonia,
Okay.
in Polysiphonia also specialized trichoblast, specialized
yes.
branch there.
Yes.
In specialized branch, spermatangia developed. From the spermatangia, spermatia, non-motile. Another important difference between Oedogonium and Sargassum and Polysiphonia. Oedogonium and Polysiphonia both mobile gametes, but in Polysiphonia non-mobile. They are called as a spermatia.
Okay.
And another important one is, if you take Polysiphonia female reproductive organ, the special elongated neck present, that is called trichogynum. And inside the swollen structure, egg cell present, and the spermatia reach to the egg through the neck cell, through the neck, and as a result final formation as a zygote.
Yes.
So zygote formation similar in all types, but the difference is
little
formation of
advanced sexual reproduction system.
Right.
We are looking in rhodophytic member.
Right.
Okay,
The difference is formation of sexual reproduction organs.
Yeah,
That is the
okay.
difference.
Thank you, sir. You are given the nice, excellent. So that Oedogonium and Sargassum and Polysiphonia. So that is often reproduction system or life history, sir. So very good. Thank you very much.
Yeah.